I would like to welcome Professor Adam Godzik, and he's Professor and Program Director, Bioinformatics and Systems Biology Program at Stanford Burnham Medical Research Institute, and he has come all the way from La Jolla, California. Thank you very much.
Okay, thank you very much. It's a great conference, great to be here. Thank you very much for the invitation. I want to keep it short because I understand we're running behind. Okay, I just put in my timer so it's not run over. So I'd like to tell you a little bit about an effort I did recently in my group where we tried to marry systems biology and structure to arrive at, somehow combine insights coming from these two methods. So recently systems biology became a buzzword, and a lot of, it's like a new field which started, and it's often contrasted to standard biology. And in the sort of standard biology, when we study a system, we look at a very complex system, and then for one reason or another select one element of this, for instance a protein, and then we isolate it, and it allows us to control it very well. So we can do experiments, and we can study it very deeply, and then if we try to understand how this element would tell us about the original program, we have to sort of map it back. And of course this is a very different conference because almost every single talk that deals with high throughput and large scale information, but I was recently on a meeting in my home institute, and I noticed that all talks can be basically mapped to a very simple, all titles of all talks have the same system: name of the gene in name of the disease. So at least in people I talk to, this model really dominates. So in system biology, we try to approach a complex system from a different way. We look at high throughput techniques which provide us information about all elements of the system. Then we try to combine all this information to build a network model, and then we try to simulate this model to predict something about the system. So as I said, most of the talks over the last few days here we're dealing with something which belongs more to this group. And of course, advantage of this is that it can be applied to very complex problems, including those which have sort of multiple causes. On the other hand, this works well only on a single cause problem. So if you have one protein which really is important for something, you can discover and analyze it this way, but it very often fails. So when you collect all this information, you get this kind of very complicated maps, which describe relations between proteins. And however, it's very complicated, but they're realistic and sometimes predictive models, and there are several different successes in systems biology where people really have built models which are really useful. They provide detailed prediction of how a system like this would behave. However, when I was trying to learn something about systems biology and did some baby steps there, I noticed something strange, that all such models I have seen so far are what I call flat. So even if proteins appear there, there's a point, a name, a symbol. But in reality, all these proteins and all these elements in this network have three-dimensional shapes. And these shapes are very important for the function. So the question I asked myself is, how can I improve understanding of this network by somehow bringing in information about three-dimensional shapes of elements of this network? And it sort of helped that I was a theoretical computational biologist, but I deal a lot with structures, and this is just one slide summary of something which you probably all know, but I want to repeat it because it's so unusual in the context of genomics, that proteins actually have structures. They have unique three-dimensional structures, and on top of this, especially human proteins often contain multiple modules. So if you look along the protein, you can cut it in one place, and this part becomes almost independent protein. So on average, human proteins have 2.3 modules, but especially proteins involved in regulation, and most of proteins like this are exactly the ones which are involved in diseases of multiple semi-independent entities, domains. And each of these domains have very often independent structures, so if we cut it off, it would solve on it, or it would fold on its own and would behave well in solution. And very often it has independent function. So this, for instance, is an example of a sugar hydrolase or carbohydrate hydrolase. And it has two functions. One is binding a substrate, another one is catalytic. So in some sense, you can factorize the function into two parts. And you can take this domain, move it somewhere else to another protein, and it will still bind carbohydrates. You can take this protein, you can merge it with another domain, and it will suddenly change specificity, because this domain defines specificity, and this domain defines enzymatic function. And it seems trivial, but especially in the end of my talk, I would come back to how we can use this insight to understand better networks, and especially disease networks. So important thing to remember is that at this point, we know structures of about 85,000 proteins. And of course, any talk on genomics, you would realize that it's immediately dwarfed by hundreds of millions of sequences we know today. So one can wonder how we can use information about 80,000 proteins to understand networks, which in principle can describe organisms which were never studied by structural biology. So this is a question which I wanted to bring. So what can we gain by taking a model of a network? This is a simulation or network model, which I would come back to in a moment. And what can we learn by putting a structure in each point? Okay, this was supposed to be a dynamic question. Okay, so when we come back to this comparison between 85,000 structures and 100 million sequences, the answer is yes, we know very few structures experimentally. But what helps us a lot is that protein structure is relatively conserved. So if we have a close homologue of a protein with a known structure, we can with high probability say that structures are almost identical. And when you do this, you realize that somewhere like 15% proteins in eukaryotes and around 25 proteins in bacteria can be very accurately modeled. So by accurately, I mean that it's basically as accurate as experimental error. So for these proteins, we really know what the structure is. And this is not very encouraging, because it means that if we build a model of an organism, on average, 15% of the genes can be modeled. So our structural systems biology dies before it starts, because if you can model only 15% of nodes on the network, you can't really do very much. But this is misleading in the sense that the word accurately here is highlighted. These are proteins we can really model very well. But things change if we allow tools for more distant modeling and what is called fault recognition, where we can very often, the boundary between known and not known is not so sharp. So these are things that are very well known, and then I think just sort of very well known, somewhat known, and what it means is that if we go along and allow models to be somewhat less accurate, we can actually extend this 15% to much, much farther. On the other hand, we have transmembrane proteins which have their own roles, and then we have proteins such as disordered proteins. So this completely unknown region of protein universe is actually quite small. So my estimate is somewhere between 10 and 15% of all proteins. And of course, once we move into this territory, we have to be worried about the quality of our model. And this is something we should, admit upright, that once you move from this area to this area, the model would have significant errors. On the other hand, you still have quite a good idea of how it looks like. You can assign a fold, you would know it's a beta sheet or beta protein or an alpha protein, and at least for some applications, models of quality from this area can be very useful. So with all this in mind, we decided to go and try to approach a small model system with these ideas. And what helped us a lot is that all this thinking was happening in the context of the high throughput structure determination project, which I was involved with early on, and a few years ago, NIH decided to fund or help develop automated structure determination pipelines. And that was part of a system like this called Joint Center for Structural Genomics. And over several years, we have been developing automation for all the steps in protein structure determination. And actually, I was not developing this, I was developing the database which collected all the data. It was a massive data management exercise. And we developed this JCHG database which collected information from all the steps. But as a part of it, we have learned how to solve protein structures very quickly. And in this project, our center alone solved around 1,400 protein structures. And between us and other structural genomic centers, we solved about 12,000. So we can show pages and pages like structures like this, and if we approach structural biology in the standard way, each of these little proteins is a story. We can go in and study individual positions, but from my point of view, I was interested in how many of these proteins I can use to put on my network model and get this global structural picture of the biological network. So I think this slide sort of belongs a bit later, but when I was showing this kind of pictures of proteins in these rows and columns, I was struck by a similarity between this and pictures from the catalog of parts, where things are ordered by the number or by something else, and they don't really make much sense. So if you look at things like this, we still don't know what they do and why. And I thought that what we need to make sure sense of the structures, to put them in the context, where like in this case, when you show parts in a functional context, you would understand better why they get the shapes they do. So this is the example of how we could do it in a warehouse situation where suddenly you understand that it's a gasket and is round because it connects these two things. So what would be a context in which you can bring structures in the same way to make sense? So we thought that this is not a random collection of parts, they form a self-sustaining organism, so that the context would also help us to understand structures. So in this sense, we believe that when we combine structures and network models, it would help us to understand both. So this is what we came up with this funny name, structural systems biology, and started to see if we can implement it on a model system. And the model system we chose to work with is a small bacteria called Thermatoga maritima. It's a thermophilic bacteria, one of very few. Most of thermophilic organisms people study are archaea. It has carried the name because of unusual, very rigid second member. So technically it's a gram-negative organism, but in most gram-negatives this outer membrane is very, very thin and very fluid. In this particular organism is very rigid. So people thought it's a toga, like a Roman cloak, and they called Thermatoga. So sequence was a second organism sequenced, a second bacteria sequenced by Tiger in 1999. It has 1877 official ORFs. I put this official of highlighting it because, as always, it turned out that it had a few more, and we actually discovered some of them by analyzing the genome very accurately. And very unfortunately for us, and I would get to this later, it's not a pathogen. So NIH was not very happy that we studied something like this. But recently there is some growing interest in Thermatogallis, and perhaps this would be a good place to talk about it because they are heavily involved in energy production, and some of them are known to essentially infest all oil fields. So I understand that there are sort of pathogens of oil fields. If you like people can get infected, also oil fields can get infected by Thermatogallis, and some of them actually start releasing hydrogen and lead to explosions. So apparently there's some growing interest in this, but honestly I don't know too much about it. Anyway, it was picked up in a very picturesque place in Italy, near Vulcano Island, and it's living in a shallow sediment. So it's not a deep sea vent, it's a kind of very shallow sediment. So, as I said, the genome has 1,800 ORFs. We solved about 200 structures from this organism. Still not enough to really build a good model. Some others were solved by other groups, so together it's one of the best covered organisms, but as you see, this number is still much shorter, much smaller than number of ORFs. But what helped us a lot, as I mentioned earlier, the number of structures, actual structures in the organism is very high. But when we add things we can model or we can predict, including fault predictions, we're getting close to the number of all the proteins which can possibly solve from this organism. And at this point we stand about 100 proteins short from a complete coverage. And of course this is for entire organism, and what we decided to do is to try to build a model of the metabolic network Thermatoga. So how to build a network model? So it's an ultimate exercise in integration. We just went to literature and started collecting all the information about this organism we could find, picked up functions of all the genes, and started to sort of put them together and merge them into a working network where we could connect all the points and explain all the functions. So this is a project mostly done by Bernard Paulson Lab, who pioneered this kind of modeling. And they tested a lot of these methods on E. coli, which is like the highlight of the best model system they worked with. So we basically learned from them and together used this same approach to Thermatoga. So the first 300 reactions and genes were very easy. They were well conserved between organisms. We can easily put them together. The only problem is that network built from this 300 didn't make any sense. There were holes. There were things which we knew this organism is doing, but our model didn't. And then we used iterative evaluation. So we built a model, we tested it, whether it works. It was missing something. We used the old method pioneered by, best described by Conan Doyle, that we basically try to find the best possible solution when we eliminated everything else. And after a few weeks or months of work, we got our model. So when we had the model, we started validating it, and we found that it really describes everything which is experimentally known about Dermatoga maritima. So with our model, we can describe its evolution, its behavior in all the experimental states it was known. It would die. With food, we know bacteria would die and would survive and produce, including hydrogen in specific situations. So we believe that this model at this point summarizes everything which is known about Dermatoga maritima metabolism. And then we started looking at this 478 genes coding for 478 proteins, and we realized that we have basically complete coverage. We have either models or experimental structures for all of them. So in this case, we achieved our goal. We had a working model which describes at least metabolism of the organism, and we had experimental or model structures for all the network, all the nodes on the network. And of course, something which is very important to remember is that our model covered 478 genes, so 1,398 were not in the model. So there's still a lot about Dermatoga we don't know. So in some sense, we can, it's a typical problem in all the modeling, we can model all that is known about Dermatoga, and we still left over with 1,300 genes. We have no idea what they're doing. So in our hands, they are not necessary. Of course, Dermatoga is not stupid. It carries these genes for a reason and just tells us how little we know about the functions of these other genes. So we call it a first-generation model. And again, this was published some time ago. I gave a talk about this when I was here a few months ago, so I will just show you one slide of what we have learned from this model. And because we knew that quality of the models were not very good, we knew we couldn't, for instance, describe metabolism, we couldn't do docking, so we focused on evolution of the network. So one of the questions we asked is, for instance, how does network have evolved? And we could compare it by looking at similarity between structures and position on the network. So we asked the question, if two proteins have similar function, or so if they are very close to the network, how likely it is that they would have similar functions? And again, this is a question we can answer even if our models were not very good, because even if there were some errors, we could still tell that these two proteins are more similar to each other than to something else. And what we have learned was quite surprising, is that enzymes catalyzing similar reactions are much more likely to have the same fault than enzymes catalyzing adjacent reactions. So basically, there are two models of how metabolic networks can evolve. One of them is that there's an ancient enzyme which catalyzes a lot of reactions, and then it gets specialized and starts catalyzing individual reactions, but this would mean that all the neighboring enzymes should be similar. And there are some pathways which do this, but overall, the model of grabbing something which is useful and sticking it in the middle of the pathway wins by about 1,000 to 1. However, this connected model still is much higher than unrelated. So this model of enzymes splitting and dividing and then specializing still accounts for much more than just random. So these are the kind of questions we could answer with this first generation model, where some of the models, as I said, were not very good. So later we thought that Dermatoga is somewhat obscure organism. Not many people are studying it, so we may be more lucky when we go back to E. coli. So we did the same experiment in E. coli for K12, the laboratory strain. Again, we modeled the entire metabolism. We actually used the model, which I said is a flagship model of Bernard Paulson's group, and we managed to model about 90% of the whole network. And what is else, because the E. coli is such a well-studied organism, we managed in most cases to model enzymes both in an upper and a hollow state. So for every node, not only we had a model, we had two models of how this enzyme looks like with the substrate and without the substrate. And this allowed us to estimate how this reaction would depend on temperature. mostly because the larger the structural change between two states, the more likely this enzyme is likely to be affected by temperature. So we predicted this thermostability of most of the enzymes in the network, and then we can answer a very abstract question, which is basically why E. coli is not a thermophile. And of course we came from Dermatoga, so we are used to thermophilic bacteria, but we sort of, it's a very naive question. And E. coli is not a thermophile. When you heat it to about 42 degrees, it basically dies. But what we discovered is that when we looked at how enzymes affected by temperature the most, where they are distributed in the network, we have realized that they grouped in some very specific pathways. So they were not distributed uniformly around the entire network. Most of enzymes in E. coli were pretty thermostable, and they concentrated in just few pathways. So you thought, what happens if we take substrates of these pathways and add them to the media? Because then E. coli would not be affected by the fact that these enzymes would be denatured. And what had happened, we actually can rescue it. By adding the mixture, we can grow E. coli quite effectively in 42 degrees centigrade. So we managed to turn E. coli into at least limited thermophile. And this was a very specific prediction we made. And it was also quite interesting that completely coincidentally, we didn't know about this when this project was happening, there was a group which was interested in directed evolution of thermophilic E. coli. They basically put the E. coli into a vat and start growing it and slowly raising temperature and waited several years to see how mutations would help it to accommodate. And then they sequenced it. And we approached them to get the sequences, and it was quite interesting that most of the mutations discovered in this artificially evolving thermophilic E. coli happened in the enzymes which were predicted to be least thermostable. So again, it was published this year in Science, and it gave us a lot of good feelings, because we could say that yes, bringing structures to the network can actually allow us to predict things which were not built into the network. Nobody ever studied questions which are so irrelevant to medicine, like why E. coli is not thermostable. But as you all noticed, this is the Genomic Medicine Conference, and when we were engaging in experiments like this, we were sort of gently reminded that we are funded by NIH, which is National Institute of Health, and not National Institute of Dermatoga Maritima. And unfortunately for us, neither Dermatoga Maritima or K12 is a pathogen. So basically we said, well, all these tools are nice, all these insights are very interesting. Can we use it somehow to some questions related to disease? And what helped here is that I work in the institute which was formerly known as La Coya Cancer Research Foundation, and 90% of my colleagues work on cancer. And they were sort of poking me all the time, nice talks, very nice. Who cares about Dermatoga Maritima? Can you tell us something which will be useful to us? So when I talked to them, they told me, well, look, there are some databases recently released, such as, for instance, Cancer Cell Line Encyclopedia, Genomics and Drug Sensitivity in Cancer database, and they give a lot of data. They give a lot of data about proteins, relations with proteins. Can you use this kind of insight to help us understand something about this data other people have not? So we focused on this Cancer Cell Line Encyclopedia. And again, this is published paper in Nature. They tested 427 cancer cell lines. They ran them against 24 anti-cancer compounds. They sequenced all these cell lines and identified mutations in about 1,700 genes. And they classified them all by how they react to these drugs. So that they basically checked whether given cell lines are more or less sensitive to this cancer drug. And this is all released publicly. And then they analyzed this data and identified multiple mutations and gene biomarkers in cancer response to drugs. So they found that certain genes, when mutated, would increase sensitivity of this cell line to this given drug, or would decrease it. But well, what can we gain if we come in with our understanding of structures and try to analyze this data? So if you look at this analysis, it was clear that they had two problems. So, of course, we know that some specific mutations are very important, but in this particular statistics, these mutations were extremely spread out. So out of about 42,000 mutations they identified, only 600 were seen in more than two, actually that's a mistake, more than two different cell lines. So basically all mutations were unique. So there are only 2 or 3 mutations, individual mutations, which they identify as biomarkers of sensitivity. So what they did, they switched back to gene level, which is a usual method of analysis. And even if I hear talks given here, everybody talks about genes like pro-cancer genes, anti-cancer genes, gene biomarkers. So it's obvious that in genomics, not surprisingly, a gene is a unit, which everybody talks about. But if you look at the genes, as we discussed it earlier, codes for proteins, and proteins have domains, you can naturally look back on the gene and say, well, there are domains in genes, or regions. Regions in this gene code for this domain, region in that gene code for another domain. So could we learn something from it? And obviously we know that not all mutations are the same. Right? So there are very specific, well-known cases where identical mutations in the same protein would have opposite effect. So for instance, there's a mutation which gives resistance to this drug. On the other hand, it increases sensitivity. So we said, well, can we gain something by bringing structure to the picture and look at all this data, remembering that genes are not ultimate units. We can sort of increase the sensitivity or the accuracy of our approach and get somewhat halfway between these mutations which are too spread out and genes which possibly are too big. So by doing this, we found a lot of interesting things. It sort of worked. So the first thing we noticed is that there were some genes, like this one, where if you look at the gene level, entire gene, there was a signal there, but was drowning in the random number of mutations which didn't make any effect. So if you look at the whole gene correlation with sensitivity to a drug, it would not be statistically significant. But this gene has 2 domains, and when we separately ask the question, is this domain related, or mutations in this domain are related to sensitivity to the drug of this, there was a dramatically different answer. So in this case, mutations in the kinase domain were below the threshold of significance, and we can say, yes, they are related to the sensitivity to the drug. And basically, unrelated mutations in this other domain was shifting it away and basically drown the signal. We saw some other examples where for instance, genes, again, the same thing. We can look at separately a receptor domain in other regions and see that some connections, some relations were similar, statistically significant, others were not. In other cases, we found examples where original genes were not found to be related, not to be significantly related to drug sensitivity, because different domains were responsible for answers to different drugs. Again, when we separated them into domains, we could read these signals carefully. We had some other cases where the same gene and the same drug, but there was a different effect. A mutation in one domain was making it more sensitive, a mutation in another domain was making it less sensitive. And again, it was a reason why the gene as a whole, when analyzed as a whole unit, didn't give any signal. And in all these cases, we also did the full modeling of 3D structures, and we get some other information from there. But at this point, I just want to focus on this first step, where we actually just divided cell into proteins into domains. And what we also noticed is that we use domains which are known, the databases of domains, and a lot of domains in human genomes are not recognized. So there are a lot of proteins which seem not to have any domains, which doesn't mean that they don't, we just don't know them. So we made some effort in a related project where we started to predict new domains in the human genome. And we have a database of these putative domains, which nobody had proven. And we found for many of them, again, we can see a difference and actually significant correlation to drug sensitivity in these putative domains. Even that officially they don't exist, but when we put them into the picture, we can see that they differ from the neighbors in the pattern of responsiveness to drugs. So in summary, we believe that yes, three-dimensional structure information can bring new insights in understanding cellular networks. We just started working on one particular example of a network, which is a cancer network. We haven't really used all the three-dimensional information. We're doing it now, but we just use the information, the domains, that proteins are divided into domains, and we already got in this simple experiment, we discovered about 170 new markers correlating response of cells to drugs, and there are also other disease networks. We are currently, for instance, working on a model of immune response to viral and bacterial pathogens using the same language. And if anybody has an interesting data set we can look at, we would be happy to try our approach and see if we can see something which was missed in this normal approach. And I don't think many people know what Stanford Brennan Medical Research Institute is. We have very famous neighbors, though. We are sort of nestled between UCSD, Salk, and Scripps, all in this little square mile of research institutes in La Jolla, California, very close to the sea, which makes us sort of similar to Jeddah, but we're a little bit closer to the sea. So actually, you can walk to the sea from there, and beach and surfing. And we also have medical school, graduate school in integrated biology. We actually apply, accept applications, and January 14 is the next deadline. We have postdoctoral positions available, so if anybody's interested, let me know. And of course, acknowledgements. So the Tematoga work was done by Ying Zhang and Ines Thiele, who's now a professor. Actually, both have now faculty positions. She's in Rhode Island, and she's in Reykjavik. Domain biomarkers was done by Edward, and Janwen and Łukasz are my modeling and fault assignment experts. And of course, more than half of this work was done in Bernard Paulson group at UCSD Systems Biology, and Roger was responsible for the E. coli project, and Ian and the JCSG team for all the structures and work on the. Thank you very much.